Mini Exam 41A, Mutations by Callie Cole. Okay, so the prompt gave us a normal protein sequence with its amino acids and a mutant protein sequence with its amino acids, and it wanted us to figure out how you would translate the mutant proteins into their RNA sequence and whether or not it was an insertion or deletion that caused this mutation. So the first step in figuring this out would be to look at the normal proteins that they gave you and figure out their RNA sequence. So each amino acid for the protein has a codon sequence, which is a 3 nucleotide sequence that codes for that particular amino acid. And so you just go down the line and you label each one of the amino acids with their proper codon. And then in some cases like this and this, you can see there are multiple letters because they're multiple nucleotide sequences that could code for the same amino acid. And so you want to label all of those because you have to figure out which one of these sequences you would actually be using. And the same goes for the mutant protein down here. In a couple cases, the same way. You don't know which one you're going to be using, and we're about to figure that out. So the first step after you've written out all the possible sequences would be to see where the difference occurs. So A match, UU, GG, and so on and so forth. Our difference occurs here in our third set, the U and the A. And so you're trying to figure out if it's an insertion or deletion. So if it was a deletion, then that U would be gone, and the next letter would need to be A to match up with this A here, but it's a U, so that doesn't work. If it was an insertion, which it is in this case, it would be added to the mutant, and the next letter would need to be that first U, and it is. And then the second one would also need to be U, and we have 3 options, and that's how you figure out which of the 3 you need. In this case, it's the U. And then the next option, you can have a U or a C. We need a C, so you would pick that, and you continue going down the line until they're all completed. And then once they're all completed, you can see our mutant RNA sequence down here as follows. And it would be due to insertion of this single amino acid here, which creates a frame shift. And basically it rearranges the reading frame of the amino acids, which is what creates the truncated non-functional mutant protein line here. It stops and it messes up the sequence because the first 2 are the same, and then the third set is different, and because of that one additional one, all the following amino acids would be off a letter or off enough letters that it causes the actual RNA sequences to code for different proteins.